Okay, we've been talking about essentially regulatory systems that, not really even systems, regulation of single promoters. When we talked about the lac repressor and the trip repressor. Turns out the trip repressor actually does have multiple targets. The lac repressor really only has the lac operator. But in fact, a lot of regulatory systems in bacteria, you have a single regulatory, a single transcription factor, lac repressor, trip repressor, or whatever, but they actually, there are multiple different promoters that they can regulate. And so there's more than one of these molecules floating around in the cell, and they are able to coordinate to regulate whole sets of genes that can be in multiple places on the chromosome. So this is what we call global gene regulation. The other term I want you to note here is the idea of a regulon. So the regulon is the whole set of genes whose expression is coordinated to respond to the same environmental signals through the same regulatory proteins. All right, so we're no longer talking about just one promoter and one operator, but multiple promoters and multiple operators, all of which are targeted by the same transcription factor. And that transcription factor may be responding to one or multiple signals. So catabolite repression is actually a good example of this. Even though the lac repressor really only has one target in the cell, the cap protein has a bunch of targets. There are a number of different metabolic pathways for the catabolism or the breakdown of different sugars that are controlled by whether or not glucose is being used. Now, your book talks a lot about another one of the SOS response. We're not actually going to talk about the SOS response. It's fairly complicated, and although it's very interesting and useful, I decided there was other things that I would rather talk about than the SOS response. So let's talk about some other global gene regulatory systems. And so what I want to talk about here is let me make sure this is the right slide, yeah, two-component regulatory systems. So there are a lot of different versions of this in just about every bacteria. E. coli has, 20 to 40 of these present. And so what I'm going to talk about first here is just the sort of the framework of what the two-component system looks like, and then we can talk about how it's implemented. Okay, so essentially when we have the two components are this membrane-bound component, which is also called a histidine protein kinase or a sensor kinase, right? So it's located in the cytoplasmic membrane of the cell. Now this could be gram-negative or gram-positive, but it's located in the cytoplasmic membrane, not the outer membrane. Okay, so the cell wall is out here, and then if there is an outer membrane, it would be out here, right? But this sensing domain is located on the outer face of the cytoplasmic membrane. And then inside the cell, so there's a transmembrane domain here, is the so-called transmitter domain. This is the kinase domain. Okay, so whatever our signal is here, it may be the presence of some nutrient or other small molecule that's significant to the cell in some way so that it wants to respond to it. Okay, when this effector molecule, you can think of this as the effector like we were talking about in the last lecture, when it binds to the sensing domain, that information is transmitted to the inside of the cell, and then this kinase domain becomes active. So the first thing it does is it autophosphorylates, meaning it takes a phosphate group from ATP and puts it onto a histidine residue. That's why it's called a histidine protein kinase, okay? Then that phosphate is going to be transferred onto, the receiver domain of the response regulator. So this is the second component, the response regulator. So you have the sensor kinase and the response regulator. So that phosphate gets transferred onto this receiver domain, and then most of these are transcription factors. So that allows it to go and bind to the DNA and do its job. Now its job might be to help transcription or to hinder transcription. In the case we're showing here, it's going to help transcription, so it's going to be an activator. It's going to help this RNA polymerase initiate transcription of the target gene here. But really, whether it serves as an activator or repressor depends on where the operator sequence is located relative to the RNA polymerase. Okay, so signal from the outside of the cell, the sensor kinase is in the membrane, senses that signal, communicates it into the cell, phosphorylates, transfers the phosphate on the response regulator, and the response regulator does its job. Okay, so there are lots of different versions of this, as I said, right? So these are lots of different bacteria. Agrobacterium is one we're going to show you in just a second, and it regulates the ability to form tumors in plants. This is another soil bacteria that controls nitrogen fixation, or that carries out nitrogen fixation, Cynorhizobium melliloti. And these dicarboxylates are actually the food that it gets from its plant that it is in a symbiotic relationship with, control of phosphate metabolism, oxygen detection, etc. And then down here, virulence, which is something we'll talk about more in the lab. Okay, so the point is that there are lots of different ways that this two-component system can be used, and in doing so, it will respond, to lots of different signals, right? So the trick is, which signal is it going to respond to? And that's based on what molecules this sensor domain can bind to. And then the sensor kinase domain is going to have evolved to interact with the different response regulators, right? So that each one of these can carry out a unique signal transduction process inside the cell. This is the example I told you we talk about. Agrobacterium tumefaciens is a bacterium that infects plant tissue. It infects root tissue, and it induces the plant cells to grow and to feed it, right? So it essentially is like a bacterium that's causing cancer in this region of the plant that it has infected to create what's called a crown gall. Okay, so what is this sensing here is molecules that are released by the plant, by damaged plant cells. Okay, so when it senses, oh, there's damage in this plant, this might be an opportunity for me to invade it. Those molecules are bound here, and that is communicated to the interface, the kinase domain. That kinase domain that phosphorylates this VIR-G protein. This VIR-G protein then goes and turns on RNA polymerase. These VIR-A, the sensor kinase, and VIR-G, the response regulator, are encoded on a large plasmid that's present in these agrobacterium cells. Okay, those are the players in the regulatory system. These virulence genes are what is whose transcription is turned on when VIR-G is activated. What's going to then go on to happen is this agrobacterium is going to express proteins that are going to allow it to invade the plant tissue and cause the disease state, which is going to then support the growth of the bacterium. Okay, so that's the virulence, is the set of proteins that are involved in being able to invade the wounded plant and induce plant cells to grow to feed the agrobacterium, right? So different two-component systems allow microbes to respond to various environmental stimuli, right? So the pairing between the kinases and the response regulator determines sort of the specificity of the system. So, in each different system, the differences between them have to do with the, what signals they're responding to and the sensor domain of the protein kinase, right, out here. And then what response regulators are going to bind to. And then finally down here, what specific operator sequences those response regulators are going to bind to on the DNA, which is going to control what genes they transcribe. So there's multiple opportunities for evolution to differentiate these two component systems. At the same time, there occasionally is what's called crosstalk between these, where, for example, a kinase will inadvertently phosphorylate a different response regulator because it's similar enough to the one that it's supposed to be binding to, right? So that crosstalk can actually affect what goes on in the cells in some cases. Okay. Now we're going to talk for a couple minutes now to wrap up this segment about chemotaxis. And you might be wondering, why are we talking about chemotaxis? Because everything else we've been talking about is regulation of gene expression. Well, the reason is that the components, key components of chemotaxis are actually related to two component systems. There's a kinase and a response regulator. They're just not connected to regulation of gene expression. Okay, so just reminding you about what chemotaxis is basically controlling the rotation of the bacterial flagellum to move towards an attractant or away from a repellent. This image is just to show you that there are ways that we can there are ways we can assay that and that we can genetically manipulate that. Okay, so this is what's called a capillary assay. You put some molecule that you want cells to be attracted to at the end of this capillary here, and then stick it into a test tube or a flask that has the bacteria in it, right? Now this has to be like continuously filled with fluid, so you end up creating a gradient where your attractant is up here, and the cells that can sense it and move towards it move into the capillary, but the ones that can't stay out here, right? So this is sort of a way of separating the ones that can respond and the ones that can't respond. And so what's been done in Salmonella and E. coli, for example, is to induce mutations by chemical treatment or UV treatment in the bacteria, and then look for mutants that can't chemotax anymore, so they would stay out here. And there's different ways this is done, but the bottom line is mutations in genes involved in chemotaxis have been isolated. So this is just giving you a little insight into the process by which this system was sorted out. Okay, so here's the way it actually works. And this is a little complicated, so we'll go over it in this slide and then one or two other slides, right? The sensor in the chemotaxis system is not the histidine kinase, not the sensor kinase. It's a separate protein that has a sensing domain out here to bind to attractants or repellents. And then, like a sensor kinase, it communicates that information into the cell. Now, the sensor kinase, or the homolog of the sensor kinase in the chemotaxis system, is this protein Ka. Again, it is not directly sensing attractants or repellents. It is sensing the response of MCP to those attractants and repellents. MCP, as you'll see in a subsequent slide, stands for methyl accepting chemotaxis protein, right? You'll see that term again in the subsequent slide, so we don't need to worry about it, what it means right now. Okay, so the sensor kinase here phosphorylates, and then the phosphate group is transferred to a response regulator, the Ky protein. So Ky without phosphate, Ky with phosphate. Phosphorylated Ky binds to the flagellar motor and induces it to turn in the clockwise direction, which in E. coli and Salmonella causes the cells to tumble. Okay, so tracing this back to the start here, if there's no attractant bound, Ka is stimulated to phosphorylate. So if there's nothing there, it's stimulated to phosphorylate. Ka then transfers phosphate to Ky, which goes here and tells it to tumble. Okay, so if there's nothing that it's sensing, it says, hey, let me look another direction. So the cell tumbles, and then it runs for a little while. Okay, and it does that because there is this phosphate group on Ky is very unstable. Okay, so it is rapidly removed, right? So it induces a tumbling, it's dephosphorylated, falls off, and then the flagellum rotates counterclockwise again, so it runs. Okay, so the bacteria don't spend a lot of their time tumbling. It's just a way of reorienting them. They spend more of their time running. Okay, so anyway, we have key A and key Y here, right? Now, how do we get it to run in the right direction if it is sensing something? Well, if MCP is bound by an attractant, then MCP does not stimulate key A to phosphorylate. Okay, so there's less of the phosphorylated version, which means there's less of the phosphorylated key Y, which means that the bacterial, the flagellar motor keeps turning counterclockwise and keeps running. Okay, so basically the attractant binding keeps key A from phosphorylating key Y, and so there's no key Y phosphate to tell the cell to tumble. Okay, now how does, let's see, all right, probably should have put this slide up front, but now that you've seen different players, here are the rest of the players. Okay, we already talked about key A and key Y. Now we have key R, which methylates MCPs, key B, which demethylates MCPs. Key Z you already saw because it dephosphorylates key Y. And then key W is sort of an adapter that helps MCP bind to key A. Okay, so these are the players. So let's talk then briefly about what these other ones do. What's going on with the methylation? So I told you that MCP stands for methyl accepting chemotaxis protein. So that methyl comes from key R, but it can be removed by key B. Okay, so why is that important? Well, here we go. You have two sides here when there's not much attractant and when there's lots of attractant. Okay, so if the cell is moving up the gradient, the system is being pushed this direction. So key A is not phosphorylating, key Y is non-phosphorylated, and the flagellum is moving counterclockwise. Right. So these are the things I already told you. Well, what is the role of the key B and key R, right? The methylation. So essentially methylation by key R makes the MCP less sensitive to binding of attractants. Okay, so as you bind to the attractant and you keep running, the only way for the cell to know whether or not it's going up a gradient is if your only two options are bound and unbound, then you have sort of a threshold level, right? But if you want to keep going up a gradient, then once you become bound, there's no further sensitivity unless you make it harder to bind so that you need even more of the attractant there. Okay, so as you get a lot of the attractant bound to the MCPs, this key R adds more methyl groups and it reduces the affinity for the attractant, reduces the MCP's affinity for the attractant, right? So you have to have more and more attractant to stay bound as you get more and more methyl groups, right? So there's actually multiple sites of methylation. Okay, so key R, as long as the attractant is bound, key R will keep loading up the methyl groups to make it harder and harder for the attractant to bind. So you keep having to go to higher and higher concentration to keep it bound, okay? And eventually, the idea is that the cell will keep working its way towards higher and higher attractant concentrations, right? But eventually, if you turn the wrong way, right, then, or if the attractant goes away, then you need to reset your MCP. And so that resetting is done by removing the methyl groups that key R has added. Okay, so that's what key B is for. So essentially, key R and key B maintain a dynamic equilibrium that allows the MCP to respond to a wide dynamic range of attractant concentrations, right? So rather than just having one set point, the set point keeps changing so that the cell can sense a larger concentration gradient, okay? So at any rate, it's not just the key A and key Y, the sensor kinase and the response regulator. It's also the tuning of the sensing system, and also over here, the dephosphorylation of key Y to allow it to respond very rapidly. So all of these systems put together generate the allow chemotaxis to respond to ranges of attractants over several orders of magnitude in terms of the concentration of the attractant, right? I am going to stop there now, and then we'll come back with another video after this, okay?